So I'd like to introduce Richard Beck, who's going to talk to us about adaptive local fitness landscapes for aneuploid karyotypes.
Yeah, thank you. So today I'll be talking about maybe I should use the I have a pen. Hello? Yeah, so I'll be talking about fitness landscapes for aneuploid karyotypes today. Um, I've never really found a visualization that I've found satisfactory for fitness landscapes. So during lunch today, I just went outside and took a picture of some sand. I think it's closer than many of the fancy visualizations I've tried to put together for some of these high dimensional fitness landscapes. So that's what we're seeing there, the beach. Okay. So I'm going to begin by just giving some basic facts about aneuploidy and cancer to sort of try and motivate my talk and try and get everyone sort of on the same page as me with how I think about aneuploidy. So what is aneuploidy? Basically, it's the result of a process of missegregations or ongoing chromosomal instability. Um, it can come about in different ways, but at its most basic level, it results from errors in mitosis, which result in daughter cells receiving imbalanced numbers of chromosomes. So what effect does this have on cells? Um, so what we can see here in the middle is the copy number of chromosomes along the bottom axis of different strains of yeast. And you can see these are aneuploid, they've sort of gained different chromosomes. And at the bottom plot, you can see that the protein expression is really closely following the gain in DNA copy number. And you can imagine that this would have significant effects on the phenotype of the cells. And aneuploidy is really common in cancer. It's estimated that as many as 90% of tumors are aneuploid. And the fact that we see cancer-specific patterns of aneuploidy really suggests that cancer cells are sort of using this ability to sort of change their copy number and therefore their phenotype as a sort of substrate for selection or evolution. So how can we think about aneuploidy? How can we understand what the consequences of a specific aneuploidy, a specific pattern of aneuploidy might be? How can we make sense of this? So one way I like to think about it is to sort of split this into a dichotomy. So one approach that one could take is to take a bottom-up approach. So we could look at a particular chromosome where the copy number has changed. We could ask what genes are on that chromosome, and we can try to sort of understand at a molecular level, like what is the impact of changing the dosage of the specific genes on that chromosome. And the advantage of that, it's been done with some success by some groups in some cases. And it gives you some sort of mechanistic clarity in that you can sort of identify molecular pathways that are being dysregulated and potentially intervene. But the disadvantage is given that you're changing the dose of so many genes at a single time by changing all the genes on that chromosome, it's quite complex to try and pick apart like what is the trying to sort of disambiguate between the different doses of all those genes. So the other way you could think about it is taking a sort of top-down approach. And you can just ask the question, okay, if we change a particular chromosome, sort of not thinking about what particular genes are on that chromosome, but what impact does that have on the fitness of that particular cell or karyotype? The advantage of this is it's sort of tractable. So traditionally, if you're going to do genotype to phenotype mapping, you might have a genotype that consists of many thousands of genes. And obviously, that's a feature vector, which is very long. But if you have the karyotype, this has a length of perhaps 22. We consider autosomes. So this is much more tractable. But the disadvantage, of course, is you're sort of throwing away that information about individual genes which are on that chromosome. But I will argue sorry. I will argue that we can still gain some useful insights from this. So we can do things like look at the topology of the fitness landscapes and perhaps make some evolutionary predictions. Not sure what that was. Okay, so the method I'll present today, alpha K, just going to give a brief overview of how this works. So basically the input is longitudinal single cell karyotype counts. So the graphic at the top is just some hypothetical cell population, which is evolving in terms of its karyotype. We would take data from that. We would take the frequency of individual karyotypes. So in the images here, on the left, each row represents a karyotype, so a bunch of cells that all have the same copy number on the chromosomes. And we would look at the frequency of each karyotype as that evolves over time. That's the basic input to the AlphaK method. Here is a kind of simplified overview of how the method works. I don't really want to dive into the details too much today because I only have 15 minutes. So I'll spend more time trying to sort of suggest that it works rather than sort of go into the details. Try and give evidence that it works. And then if people are interested, obviously you can come to me afterwards for more details. So this is kind of a simplified overview, but basically we take the karyotype frequency data that we take from the evolving cell population, and we estimate the fitness, F, of each karyotype in that data using simple replicator dynamics equation. So these fitnesses, they're constant. We have a model of missegregations or mutations between karyotypes that's taken from the literature. And the output of that is fitness estimates per karyotype. So we don't actually often see a whole lot of different karyotypes in the input data. So we have a second step where we do Gaussian process regression. So you can think of that as related to or similar to a spline. And this sort of makes predictions for karyotypes that are not actually present in the input data. So we end up with predictions of fitness for thousands of karyotypes, not necessarily all directly observed in the input. Once we have these augmented fitness estimates, that is essentially our fitness landscape. There's an additional step, which is we call the cross-validation filter. So basically what we do is we take these augmented fitness estimates and we re-estimate the pipeline with one at a time. Each karyotype is sort of removed from the input data, and then we compare the fitness estimates for those removed karyotypes with the initial fitness estimates. And if we see a match, we say, okay, it's not exactly a validation, but it at least shows internal consistency of the pipeline. And we essentially discard any landscapes where this test is not passed. And this gives us our final output, which is a local fitness landscape. And you can see here in black sort of represents regions that are totally unexplored in the input data. The white is frequent karyotypes, which we observe. And then the sort of the blue sort of represents some of the inference that's done by the pipeline that's not necessarily observed in the data. So does it work? So to test that question, we did some extensive synthetic validation. And the linchpin of this is a simple stochastic agent, it's not really an agent-based model, simple stochastic simulation. So in the simulation, it's just exponential growth. It's non-spatial. Cells are just dividing with a constant rate, and that's their fitness. Into that, we put artificial fitness landscapes. We have an initial population, and we basically run that simulation, generate the output data that we need, and get our fitness landscapes. So there's two ways that we can look to try and validate these fitness landscapes. One is to sort of look at the fidelity of the landscape itself. With the synthetic data, we have the advantage that we actually know what the true ground truth fitness landscape is. So we can just make a direct comparison. And what we can also do is we can feed these landscapes back into the agent-based model. We can run the simulation and we can see if the predicted population evolution matches the sort of ground truth. So just a little bit more detail about that prediction validation, because I'm going to talk about these metrics a little bit later on. So this is sort of a basic overview of how this prediction evaluation goes on. So you can see there's three training samples here. These would be the inputs, the inference pipeline. These are supposed to represent population distributions or something like that in a low dimensional space. We'll have a holdout sample, which is not part of the prediction of the inference pipeline. And then there'll be a prediction that will be made. And there's two metrics that I've sort of come up with to sort of try and evaluate the quality of the prediction. So one I'll refer to as the angle metric. So this would be sort of the angle that the centroids of these populations make in a high dimensional space. And we have a sort of reference baseline null distribution that we can compare to and say whether we've beaten that. And then the other we call the distance metric. So we look at the distance between the holdout and the predicted population. Obviously, the smaller the better. And as a sort of baseline, we look at the distance, the longer arrow there, that the holdout sample actually evolved. So basically, this can be thought of like if our prediction for the population was just the last observation, do we at least beat that? Okay. So the results on the synthetic validation, the landscape fidelity, measured here with just a simple correlation, you can see that these values are at least above 0, so it does capture some signal. You can see that the yellow, so this is after the cross-validation filter, are much higher than the purple. And finally, you can see going left to right, as we increase the number of longitudinal samples in the data, these yellow bars are going up, so more observations, better results. In the evolutionary prediction fidelity, it's kind of similar trends as the landscape. So with the distance metric, so you're really looking down here, beating the baseline. So after the validation filter, with more longitudinal samples, we do beat that baseline at least half the time. And the angle metric here, so the is the dash line, and you can see that we're sort of to the left of that. So it's kind of working on all our metrics in the synthetic data. We also tried to validate this with some empirical data. So I guess there's kind of a lot going on in this plot. I'll try and break it down. Each point is a sample where we have a population and we have measurements on a karyotype. These are further split into in vitro lineages. So this suppressed, immortalized epithelial cell line, and this has been engineered p53 knockout to induce aneuploidy. So these are essentially successively passaged karyotypes measured successively and we get this lineage. And then we have these PDX lineage as well. So again, breast cancer cell lines, p53 mutants. And here, tumors are grown out in PDX models in mice. Tumors are harvested, implanted into the next mice. And this is our lineage. And we have samples of the tumors. So each point essentially represents one tumor grown in one mouse. So did it work on the empirical data set? So I'll start with the bad, or at least the not overwhelmingly good. So the distance metric doesn't clearly beat the baseline. So you can see here it's dependent on the lineage, but there's a couple of lineages maybe above 0.5, but some not so. But what I found quite interesting is if we look at the forked passages, so these are two sort of sibling passages with the same ancestor. And if we apply the distance metric to those passages, these don't really convincingly beat the baseline either. So what that's saying is essentially like siblings are further apart than sibling parent distance. So maybe this is inherently difficult, or perhaps the metric could be reevaluated. The angle metric, we do get good results. So predicted is well to the left of the theoretical baseline, similar to the forked. And we also can compare the angle metric on totally unrelated lineages. And nicely, we recover that that matches our theoretical null distribution quite nicely. And also total evolutionary distance, you see a positive correlation there. So that's showing that when the model predicts that the population, the karyotype distribution will evolve far, then it tends to do so. So how much time do I have? I guess quite a lot because we lost the speaker. No, it's not how it works. Okay. So I'll just finish off with a few examples of how we sort of try to leverage these fitness landscapes to get some biological insights. So in the first example, we looked at delta F, which is the change in fitness as you move from one karyotype to its immediate neighbor in the fitness landscape. So it's a sort of measure of the immediate local gradient of the fitness landscape. And we just plot here the distribution of these delta F values across all karyotypes in the landscapes. So is the in vitro, and you can see that the delta F values recovered are quite small. Then moving to PDX, you can see there's like bigger fitness effects. So what this is basically saying is that karyotype mutations have a bigger fitness delta in PDX compared to in vitro. And then when you add cisplatin treatment to the PDX lineages, then the mutation effects are even bigger. So a consequence of this could be interpreted as basically faster clone turnover between passages. So it implies a more rugged fitness landscape. And this is sort of seen if you look at the overlap coefficient between successive passages. So basically, a high value here would imply that successive passages are really similar, and vice versa. So you can see on the left, in vitro, highest values, then moving to the right, these four tend to be lower. And then if you compare within a particular lineage, or cell line at least, on treatment, so orange tends to be higher than blue. So it's most obvious down here where there's really like quite a substantial shift in karyotype population between successive passages. Second example, whole genome doubling accelerates diversification. So in our in vitro lineages, it was observed that whole genome doubling kind of spontaneously occurred. And if we plot the, the passage number on the x-axis versus the number of chromosomes altered on the y-axis for the two p53 knockout in vitro lineages, basically you can see that the whole genome doubled cells are accumulating copy number alterations much faster and to a much higher level than the whole genome doubling negative. So if we look at the fitness estimates for each of these, what we see is that for the whole genome doubling positive, the fitness is continuing to go up with each addition of a chromosomal alteration, and it's sort of plateauing perhaps for the whole genome doubling negative. And if we look at the distribution of the fitness mutations, then we see basically the whole genome doubling positive has less deleterious consequences than for the negative. And then a final example, missegregation rate or chromosomal instability itself can state evolution. So basically, to think about this, we have two different scenarios. So in scenario A, a high missegregation selects for a lower copy number. And basically, this is a consequence of the missegregation model that we use. Basically, all chromosome copies have an equal chance of missegregating. So basically, you have more of them than just intrinsically they're less stable. So you can see here, increasing the missegregation rate drives the population down to this sort of lower fitness, lower copy number fitness peak. And in the second example, scenario B, we have this sort of narrow fitness peak, which is actually higher. So all the population reside there at a low missegregation rate. But as you bump up the missegregation rate, you can see that the population spreads to this sort of broader fitness peak. And when we analyzed our landscapes sort of to look for these effects, 28 out of 35 of the landscapes showed some evidence of these effects. So that kind of implies that therapies that modulate chromosomal instability might be able to help us steer evolution of these karyotype populations. Okay. So to conclude, I think that these fitness landscapes do capture some evolutionary signal. Particularly, I think the topological analysis gives some insight into different contexts or treatment contexts, PDX versus in vitro, etc. There are a number of limitations, which I guess I'm running out of time to go into. And future directions, I think more in vitro validation So our lab's been doing some long-term evolution experiments, which I think Naomi will talk about next. So that would be a nice setting in which to try and sort of further validate the model. Oops. And it would be nice to look for ways to apply this to patient data. So obviously we have this need for these longitudinal samples, so that makes it like quite difficult, but there's certain settings or sampling techniques which I can imagine would be useful for this. So blood cancers or cell-free DNA, some exciting stuff being presented this week already that might be helpful. We have an R package, check that out. And I would just like to finish by thanking people from the clone redesign lab. So Noemi, who's helped a lot on the project, in particular Tao and Thomas, who've been sort of working with the R package and using it for some cool applications of their own, in particular Tao, and also everyone else in the lab. Thank you.
Thank you. Does anybody have any questions?
Hi. Why was it that high missegregation created broad fitness peaks? To an extent, I wasn't really able to think through that, but it almost seemed counterintuitive.
So why does high segregation select for broad fitness peaks?
Mhm.
So I guess when you have when you're a karyotype that's sitting on a broad fitness, essentially you're losing sort of copies of yourself due to migration, kind of like at the same rate as anybody else. But you're slightly benefiting from this influx from missegregations from your neighbors, and that doesn't happen to the same extent if you're sitting on a narrow fitness peak. So basically, you're sort of counting on this influx from neighbors to sort of give you that slight advantage.
Thank you.